Okay, Lab 4 with the lab on STS page. So in the lecture we talked about STS page. It's separation of proteins based on their sizes, and their charge difference is disregarded in this process because STS is added, right? STS is strong denaturing. It compromises the size differences among proteins because STS will make them uniform negative charge, because STS SO- has a strong negative charge, and STS will bind to your protein through hydrophobic interaction. We talked about that, right? So STS page is a high resolution separation technique where you can separate out your proteins based on the size differences, and then you add STS to your protein samples, 0.1%, as well as reducing agent, just like beta-mercaptoethanol or dithiothionetol, to dissolve that disulfide bridges. So sample is loaded to the wells, and the gel is set up kind of perpendicularly. Top reservoir, bottom reservoir. Each reservoir has the electrode, negative, positive, and the only way the electric current will go through is through your gel. Okay, that's how it works. And how do you determine the size of a protein? Of course, if you have size standard made up with the proteins with the known sizes, by comparing the location of your protein against those size standard proteins, you can estimate the size of a protein. Of course, that's how it's done. And the higher the gel percentage of polyacrylamide, you will have a smaller pore sizes, of course, so it'll be better for you to see the small proteins, okay? Especially the proteins that are in similar sizes can be separated out very well if you separate your proteins on the higher percentage gel. So the protein molecular weight marker, the standards you will be using for the lab, you'll be using actually two size standard markers. One is called unstained size standard marker. Just like protein samples, those proteins that are mixed up in your unstained marker would not be visible, so you will have to stain your gel to be able to see these bands. But it's made up with so many of these proteins with different sizes, the largest one being 212 kilodalton. That's a myosin protein from rabbit muscle. Smallest one is 6.5 kilodalton. It's a more bovine lung apoprotein, small protein. In between, there are so many of these proteins with different sizes. That's one marker. And another marker that you're going to use is called a pre-stained marker. Pre-stained marker means these proteins are already stained with a dye solution. So when you load your marker onto your gel, and when you separate them on a gel, they will show up as these blue bands. So that's why it's called pre-stained marker. So without your staining, you can visualize those proteins from the gel. That's the advantage. Okay. So then why do you need to use this pre-stained marker of proteins? On the other side, your protein samples are not stained, so you'll have to stain your gel anyway. The reason is, okay, I'll talk about that later on, but you'll be electroblotting your proteins from your gel to a membrane. So when you electroblotting, you want to see if the transfer of your protein is done from your gel to the membrane, okay, easily. So if these bands are visible already on your gel without staining method, when these bands are transferring from your gel to the membrane, it'll show up obviously on the membrane, right? So there's a benefit of using pre-stained marker because they will help you to validate the transfer process. We'll talk about that later again. So anyway, you will use two sets of size standard markers. One is unstained and the other one is pre-stained. And pre-stained marker has these many proteins, 1, 2, 3, 4, 5, 6, 7 proteins with these different sizes. And then the electrophoresis buffer that you will be using is made up with tris glycine. So we talked about this. Tris is a buffer, and dithiothionetol is added, and STS is added, and 0.2 molar bromophenol blue. It's a color. Oh, that's sample loading. I'm sorry. Tris glycine buffer is made of this 25 millimole tris base, and then 250 mole glycine. Glycine is electrolyte. It'll move just like electrons. 0.1% STS. And then pH is set at 8.3. That's the electrophoresis buffer. And the sample loading buffer, it has again tris, DTT dithiothionyl and SDS, and the color solution blown from below, and then glycerol. This is a dense material that will make your sample dense, heavy, so it will settle down on the bottom of the well. That's a sample loading buffer. So here's the SDS, a huge percentage. Okay. And then the gel you're going to use is already pre-made, so we purchased that from a vendor company called Invitrogen. They make and sell those gels. We'll make a gradient gel, 4 to 12%. We talked about gradient gel in the lecture. So gradient gel means it's a mixture of those two solutions. So bottom being the dense, heavy solution, and top being the light, low percentage solution. In between, there's a gradient of different concentration. So without stacking and resolving gel, we have a single gel, but since the percentage is gradient, so you will get a good separation of proteins throughout the gel. And that's the benefit of using a gradient gel. So top of the gel will be good in separating out these, proteins. Kind of large proteins relatively. And the bottom of the gel, if it's a 50%, it's good to separate out small size proteins like 10 to 43 in between. These percentage will be good to separate out different size range of the proteins, but you will have good effect throughout the gel because yours is 4 to 12%. Okay. And how do you prepare your samples? So the samples will come from your previous experiment, ion exchange chromatography. Hopefully you identified the peaks on your chromogram, and you probably match the fractions that will contain those two proteins, bovine serum albumin and equine protein. So you will hopefully try to separate out those samples. So first you will have saved some initial protein mixture sample, those two protein mixture, before you load it to the chromatography column. And then those peaks that are separated by your column. Okay, first peak fraction, another fraction containing the first peak. Second peak fraction, another fraction containing the second peak. Okay. So you'll try to see if indeed, this initial sample, the mixture of two proteins were separated by your ion exchange chromatography in these fractions or not. So you will get to see them through your SDS page. And you will run two gels. One gel you will separate out, and then you will stain to visualize those bands on the gel. Through combustible staining, and the other gel, almost identical, but that gel will not be stained. Instead, it will be electro-blotted, and then that gel will be used for your Western blot analysis for the next step. Okay, so you will each group will have two separate gels. Okay, and then how do you prepare your protein sample? So each fraction or each sample, the volume you will use will be different. Mix the calculated volume of each protein sample with the same volume of 2x sample loading buffer. That means it's a 1-to-1 ratio between the sample loading buffer and the volume of your sample that will be used. Okay. Prepare size standard by transferring 20 microliter into a new tube. So that's the size marker. You need to prepare 20 microliter, and you're going to boil it and boil the sample. So the calculated volume we'll talk about in the lab should be enough protein that will give you about 50 to 100 microgram of total protein. So based on your Bradford assays result, you should have protein concentration of your fractions or initials, and based on your calculation you will have to add enough volume to get about 50 to 100 microgram total protein amount. So it will vary based on the concentration of your fractions and initials. Okay, and once you figure it out, mix them with the 2x low sample loading dye. You will boil them together. Okay, the boiling will allow binding between SDS and your proteins through hydrophobic interaction, and cool down, and you'll load your samples to the gel. Once gel is loaded, and you'll hook up the electric current, and you'll do the gel running 50 volts for 30 minutes and then 100 volts for the additional times to get a well separation. After that's done, your gel running will be stopped, and then it will be assembled, and your gel will be stained. One gel will be stained and the other gel will be set up for your electrophoresis. So Kumasi blue staining process. Of course, you'll have staining solution ready, which contains these. Okay, Kumasi blue dye, 45% methanol, 45% water, 10% acetic acid. It'll be a thick blue solution. You place your gel in there about 2 hours through shaking. After that, you will destain in the different solution without the Kumasi blue, and you'll have to destain about 3 or 4 times in that destain solution. And after enough destaining is done, you will stop destaining and change that water, and then you will take a picture of your gel. That'll be the Kumasi blue staining. And the BIOT gel, like I said, will be used for electroblotting. Electroblotting is a fast way of transferring your proteins from the gel to a membrane. Okay, so there will be electroblotter setup at the end of the lab. I will show you how you can set up. So you will use electric current to transfer your proteins from the gel to the membrane. You will use a blotting buffer which contains tris, again glycine a little bit, and methanol, and the blotting is done within an hour, about 25 degrees and 25 volts, and it will be a very quick and efficient transfer process. Okay, you will get to do it hands-on. Alright, so that's it for the lab. I'll probably show you how things are done in the lab. Alright, thank you.